those out so you can write down your objective. The objective for today, which is Tuesday the 13th, is that students will be able to describe techniques used to manipulate DNA. Remember yesterday we talked about PCR and CRISPR, how certain genes can be isolated and amplified, and you can kind of like insert genes into your own genome, which is interesting, a little scary. So today then what we're looking at then is further in depth with biotechnology, not only looking at the DNA, but now we're going to be like cutting it up and comparing it to other people to see if you can find, missing kids or victims in a crime, all that stuff. Okay, but first we're gonna do some STAR practice. So I'm gonna give you 5 minutes. We're going to work on our do now. Like I said, the link for the class kick is already in the chat box. And of course it is posted in Teams. So you can find it in either place. I think it's posted in Teams. Yeah, it's posted there. Okay, so I'm gonna give you 5 minutes. We're working on our do now. Here we go. Hey Miguel, I'm glad you're relaxed. I'm so glad everybody is so relaxed after a test. It's so nice to see that. It's pretty great, Tim. I like that one too. Howdy, Clint. Ooh, Tati, I think likes forensic science. I think she might like this. Now remember that we will have our unit exam on Friday. So we'll have our review tomorrow and Thursday because it's a lot of information because that mock star kind of took the place of our mid-unit exam. So we'll take a 2-day review, then our multiple choice will be on Friday. There won't be an essay for this test. If you hadn't noticed, I've been making you write a lot with these exit tickets recently. It's because I was trying to make up for the fact that we won't have an essay on this exam. So the test on Friday will have 20 questions. So it's not gonna be like 50 or anything, but the 20 questions that are there, I would say half of them are easy, the other half range from medium to hard. So it's worth reviewing a little bit extra. Cool, it's so cool. Tests with 20 questions are the coolest tests. Okay, about half your time has passed. I don't think that's a fact, Will. I think the fact would be that you are there. The reason, though, feels more like an opinion. A little more than a minute left. I hope I haven't stumped you. Make sure you give me a thumbs up in the chat box when you have completed the do now. Thank you, Tim, for a very creative thumbs up. I like it. Thank you, Josiah. Remember, you give me a thumbs up in the chat box when you have finished it. Remember that we won't have an exam on Friday, and this do now will definitely show up on this test. Not necessarily this exact question, but what this question is asking. Thank you, Cheyenne. Thank you, Brandon. Oh, that's fun. I like it. It's funny because I actually had tacos for lunch. I made some green chili and I made some tacos. Delicious. I love it, you guys. Loving that action. All right. A few more seconds. Thank you, Zaria. Excellent. Okay, let's go ahead and transition over into class kick. Like I said, I hope this was not too challenging for you. If it was, don't worry, we're going over it now. And of course, we will go over it during our 2-day review, Wednesday and Thursday, before our test on Friday. So, as promised, we will be having a test over everything from this unit, going all the way back to meiosis, and that includes dihybrid and monohybrid crosses. Okay, so this is a star question, so just so you understand, these are the kind of questions you're going to get on the star about dihybrids. So let's click our highlighter first. If several pea plants with the genotype big T, big Y, are crossed with pea plants with the genotype big T, little y, what percentage of the offspring will be expected to have big T, big Y, little I allele combination? So notice this question is not telling you, hey, this is a dihybrid. They expect you to know that based on the alleles, right? Based on these genotypes. So the genotypes are telling you that you're dealing with a dihybrid problem, right? You got two traits. It's not just the one letter. It's not just big T, little t. You've got T's and Y's, right? So you got two genotypes. So that means you're dealing with a dihybrid. Well, not two genotypes. Dealing with genotypes have two alleles, specifically T and Y, which means you're dealing with a dihybrid.
So that means the percentage of the big Y, and little y will be 25%.
Well, let's go ahead and work through the problem first before we just jump into it. So what you want to do is you want to go ahead and draw your, actually you don't want to do that first. What you want to do first is do your independent assortment, right? So we've got mom and dad, right? We've got a big T, big Y, little y. We've got a big T, little y, little y. So these are the parents. So what you want to do then is you want to independently assort them into your table, right? So remember, when we're doing this, you're starting with the first letter. So big T, big Y, big T, little y. So we'll start here. Big T, big Y, big T, little y. Oops, there you go. Then you take the second letter. So you got big T, big Y, big T, little y. So you're essentially doing the same thing over again. So you got big T, big Y, big T, little y. There you go. So these are your independently assorted alleles based on your one parent's genotype. Now you got to do the same thing for the other side. You've got big T, little y, big T, little y. Oops. Big T, little y, big T, little y. Then you got your second one, little t, little y, little t, little y. And there you go. Now you have your cross set up. So if you want to go ahead and draw it or make lines and make it nice and pretty, I know that some of you guys like doing that. Yep. Yep. So the other day I was doing these Punnett squares, and I was doing it with, just the pen tool, and it just got so hard to read. I was like, you know what? If I'm having a hard time reading this, they're going to have a hard time reading this. So I thought it might be worth the effort to go ahead and just do this. Now, I was tempted at first to go ahead and put a cross here for you, like to have the table loaded up. But just in case the star doesn't do that for you, I wanted to make sure that you knew how to do it yourself. Right? Sometimes the star will have it done for you. Sometimes they won't. Okay, so let's do this thing. We got big T, big Y, little y. Boop. Big T, little y. Big T, big Y, little y. And big T, little y. And you keep going. And you start to notice a pattern, right? So we talked about this last time. Sometimes the star is going to give you problems where you don't have to do as much work as you think. So for example, on this one, the top row and the bottom row are identical because these two alleles are identical. So once you've done one, you don't really even have to do the second one. And then you look down over here, you notice the exact same thing. So here was big T, little y, big T, little y. Here you have little t, little y, little t, little y. So whatever this row ends up being, you can simply copy and paste it to the next one. So big T, big Y, little y, big T, little y, big T, big Y, little y, and big T, little y. And like I said, you don't have to do too much work. You can just copy that. And there you go. Now we have our completed dihybrid cross. So let's go ahead and take our highlighter and I guess yellow's fine. We're looking for, actually let's make it more fun. Let's use a different color. I like green. We're looking for big T, big Y, little y. So big T, big Y, little y. Big Y, little y. Then of course these are the same. You go down here. Now quickly you notice that up here we were dealing with two uppercase letters, right? Both of these alleles for the T were dominant. Whenever you come down here, it becomes heterozygous, right? There's no longer two big T's. They are big T, little t's. So none of those are going to be it. So what does that mean? That means that we have 4 out of 16 possible, right? You can reduce that down. That's going to be 1 out of 4, and that's going to equal 25%. So then A then becomes your answer. Boop. So congratulations to those of you who got it right in the chat box. Way to go. And Tim, you were so close. Instead of it being 255%, it was just 25%. That's okay, Brighton. All right, so you're due now, 25%. Now, the important thing is that you understand how to work the problems, right? Now, there's a chance that you could have done this with less work, like I said, where you can just copy and paste those 2 rows, but I highly suggest that you work through the problem anyway. Like, even though we know those are going to be the same, keep going because you can catch yourself making a mistake. You can notice something maybe you hadn't noticed before. Either way, it's always good to work thoroughly. Okay? Are there any questions about this dihybrid cross problem? I promised, like I said at the beginning of the unit, that they looked hard, but you just work through them, you do a bunch of practice, they get much, much easier. Okay? Awesome. Well, then let's go ahead and get to this fun stuff. Enough of these dihybrid crosses. Okay, so the objective, describe techniques used to manipulate DNA. Now, We're going to get to the here in a second. What I want to do first, though, is watch the video to kind of explain what's going on. If you feel really engaged by all this forensic science and you want to learn more about how this works, I included a longer video that has a little bit more explanation. The video that we're going to watch is fairly simple, and I chose it because, you just had a test. I didn't want to overload your brain. So if you want to learn more, there's a longer video, but we're just going to watch the simple one for right now. Okay, let's go ahead and jump in.
My house was broken into last week. The police can identify the culprit from blood found at the scene. How can they do that? To understand this, we need to state what is meant by DNA fingerprinting and describe some uses of DNA fingerprinting. The burglar smashed a window, leaving blood on the broken glass. Samples of blood, hair or skin can be collected from a crime scene. Forensic scientists investigate the samples in a lab. First, they extract DNA from the cells. They use enzymes to cut the DNA into shorter fragments. The fragments are placed into a well in a gel. They are separated using an electric current. Different size fragments travel different distances in the gel. The smallest fragments travel the furthest. A pattern of bands is produced. This can be compared with the patterns of DNA from possible suspects. The enzymes cut different people's DNA in different places, producing a different set of fragments. The different sets of DNA fragments will produce different patterns of bands when separated on a gel. Each person has unique DNA, which will produce a unique pattern that can be used to identify them. If two patterns match, the samples are from the same person. So, the police are able to identify the man who burgled my house using his unique DNA fingerprint. They make a DNA fingerprint by taking a sample of cells from someone and separating fragments of DNA to make a pattern. If the pattern matches a sample from a crime scene, that person was there.
Okay. So there's lots of uses for this. The most common, and I think one you might have seen on daytime TV, is like whenever they're trying to figure out who the father is, like, hey, you're the father kind of thing, right? So the results that they're looking at are actually these DNA fingerprints. So the thinking behind it is that, 99% of all people's DNA are the same, but there's that 0.1% that's different. And so whenever you're cutting up those DNA pieces, it's hypothesized, or I guess it's a fact now, that those fragments are going to line up between, similar pieces of DNA. So let's say, for example, that you got this restriction enzyme and you cut the DNA at, this spot. It's going to cut that spot at everybody's DNA. But because everybody has different, that means that DNA is going to be different across different people. And that's how you can use DNA fingerprinting to kind of, say, hey, this is your blood. You were at this crime scene, or this is your child, so on and so forth. Right. Now, there is a little bit of fallibility when it comes to it, but for the most part, it's pretty accurate. All right, so this is a virtual setup. I'm going to encourage you to kind of go through it yourself, but I am going to go through it with you so that you understand what's going on. So if I could I get a volunteer to read the crime for me?
All right, I'll do it.
Thank you, Brandon.
All right. On the evening of November 1st, at approximately 8:15 p.m., Jimmy Sweet entered his bedroom, walked over to his desk, and sat down at his computer. While reaching for the computer switch, he noticed out of the corner of his eye that one of the items on a typically well-organized shelf was out of place. Jimmy shot across the room for a closer examination. Sure enough, the object in question had indeed been disturbed. The object had been sealed in an airtight package. The package was now ripped open. The object was still inside, but it was no longer in its original condition. In Jimmy's eyes, it was now worthless. Jimmy pulled out what had been his most valued possession, his holographic NOVA lollipop. The confectionery treat was now a sticky mess. Someone had obviously
Indulged.
indulged him or herself in its sugary molecules. The lollipop's uh, holographic. Uh, what's the other one, Miss Martinez?
Image.
Image been lit away.
Very good. Thank you so much, Brandon. So here's what happened. This guy's got a lollipop, somebody licked it. So it's not just blood, right? There's all sorts of different things that have our DNA. Your hair, your saliva. It's kind of gross to think about, but yeah, the stuff that goes in the toilet. All that stuff has remnants of your DNA in it and can be used as DNA forensic evidence. But we're keeping it easy. We're keeping it, saliva. Can I get another volunteer to read the suspects for me?
Can you hear me?
I can hear you.
Okay. The prime suspects in this case are Jimmy's 7 sisters, Candy, Cookie, Sugar, Lolly, Honey, Brandy, and Carmela. Each one of these sisters is a notorious candy lover and is easily capable of committing this crime of confection. Do you want to read the rest?
Mm-hmm.
Okay. The suspects have been detained. DNA fingerprints of each are available.
Okay, you can stop there.
Oh, that's it? Okay.
That's it. Thank you, Missouri.
You're welcome.
Also, it's nice to hear from you. So what's happened is we got these suspects. So there are these sisters, and odds are one of the sisters did this, but we don't know who. So we're going to do some DNA fingerprinting and to figure out how that happened. So let's go ahead and figure out how to do that. Now, this is actually a very, very simple lab to do, and it is my hope that very soon we can do a lab like this. What happens is, let's see, step 1, add restriction enzymes to your DNA sample. What the restriction enzymes do is they cut up the DNA in specific places. So the DNA is going to be cut across everyone's DNA at the same spot, but the lengths of those pieces are going to change. All right, next we're going to pour the agarose gel. This is what's going to make like the medium that will hold everything in place. We're going to pour the DNA into that agarose gel. There'll be specific wells that will have it. Press the power button. The DNA runs. DNA has a slight charge to it, so when you run electricity through it, it'll spread across that medium. Putting a nylon membrane on top. Okay, now we're going to add some probes so we can actually see it with our eyes. Now we're going to place the X-ray on it so that we can take a picture. And here we go. Oop, so picky. Okay, so it's done. We are now going to choose the culprit. Now, this is what happens when you have your DNA run on this gel. So notice how there's pieces of all these stuff, right? Those pieces are segments of DNA. Now, we have the DNA from the lollipop, the saliva that was left behind. So I know it doesn't seem very scientific, but this is really how you do it. You literally just compare the pieces. So you look at Carmela. Doesn't look like it matches up quite right. You move over here. I see some similarities, but it's definitely not Brandy. Whoa, that looks like a pretty clear match. But just to be thorough, you go to everybody else. Not Lolly, not Sugar, not Cookie, not Candy. Honey is the exact match. So notice how we have the exact match, right? So again, the thinking is that the restriction enzyme is going to cut the DNA in specific places. If it's going to match up with the stuff left at the crime scene, then clearly it's going to be the same person's DNA. So there you go. Honey is the culprit. All right, send her to jail for looking at a lollipop, right? Okay. There's actually other really cool versions of this virtual lab, but they're much more complicated. So again, if you're interested in learning the specifics of how to do this, It's a lab that I can teach you how to do in person, but it's also, there are virtual options that I'll show you even more in depth on how this process works. Okay, so what is DNA fingerprinting?
DNA fingerprinting means you're finding like more evidence of somebody's fingerprints.
Yeah, using DNA evidence, using DNA, actually that's not evidence, let's say using DNA, actually no, I think there's probably a better definition out there. Does somebody think they can give me a pretty good definition of what DNA fingerprinting is?
Like searching like searching for like evidence.
Yeah, but I'd like to move away from evidence and focus more on the DNA itself.
Oh,
So if you want to, you can put it in the chat box or you can come off mute. Let me know, what is DNA fingerprinting? That's actually not bad, Clint. There you go. I like that a lot, Zaria. Thank you. So Zaria says DNA fingerprinting is like finding patterns in DNA. And I'd actually take off the like because it's pretty much what it is finding patterns in DNA. Very good. Excellent. Okay, so switched at birth. Although rare, there are cases of babies being switched at birth in hospitals. So what you can do is you can use DNA fingerprinting to figure out where these babies need to go. So we're going to skip all that, blah, blah, blah. What we're doing is we're comparing the bands of each set of parents to each of the babies. So in this situation, we got, let's see, couple A, B, and C, babies 1, 2, and 3. So we've got 3 couples, 3 babies. We're trying to figure out whose babies are whose. Now, in the last activity that we did, we were looking at exact DNA evidence, right? The saliva from the lollipop matched up 100% to the DNA evidence that you had based on your victims or your perpetrators. Here, though, you're dealing with parents and children. Now, think about it for a second. How can you use DNA fingerprinting to match children to parents? So yeah, somebody let me know in the chat box, how can we use the baby's DNA to find the parents? That's true, the DNA, but more specifically. Ooh, I stumped you. Oh, very good, very good. By comparing the DNA patterns from the babies to the parents, right? So even though the last time we saw the lollipop was matched directly to the person who licked it, this time we have the descendants of people, right? So remember, you are half your mom and half your dad. So it would make sense then that the DNA patterns for the child would have stuff from mom and stuff from dad, right? So it's not going to be an exact match because we're not matching, blood to a person. Here we're matching the baby's blood to the parent's blood, right? So you're going to notice that there's DNA patterns from both parents in the child. Very good, you guys. Okay, so let's do that. We've got baby 1, 2, and 3, couples A, B, and C. I'm going to give you about 5 minutes. What I want you to do is in the chat box, let me know the couples, match the couples with the babies. So tell me who, let's make it easier. Let's go 2 minutes. I'm going to give you 2 minutes to tell me baby 1's parents. All right, I'm going to put 2 minutes on the clock. What I want you to do is tell me who are baby 1's parents. Couple A, couple B, or couple C. Okay, 2 minutes. Here we go. Again, in chat box, let me know. Baby one's parents. Seeing some mention in the chat box, I like it. Again, in the chat box, let me know who are baby one's parents. You're comparing the baby's DNA to the parent's DNA. Oh, excellent, you guys. You guys are doing great. Oh, Clint, you're jumping ahead. We're just doing baby one for right now. Okay, I think I got enough in the chat box. Right, so we've got, I see this one here. I see this one here. This one here. And again, I know it doesn't seem that scientific, but this is literally how they do it. And there you go. So the couple that is baby one's parents are going to be couple, ooh, that's bad spelling. Couple B. Very good, you guys. I saw a lot of you guys put that as the answer. Fantastic work. Let's make it red. Actually, no, we're gonna make it green because we're gonna keep everything together. Okay, now I'm gonna give you a minute to do baby two. So again, baby two, I'm gonna give you a minute. Here we go. Very good, very good. Seeing some mention in the chat box. Again, let me know in the chat box, who are baby two's parents? At this point, it's 50-50 because we already eliminated one of the couples. Excellent, you guys, excellent. Great work, you guys. So then for baby two, let's make this red. That's going to be couple C. And of course, baby three, by process of elimination, is going to be couple A. Okay, explain why not all the bands in the mother's or father's profiles have a counterpart in the baby's DNA profile. What does that mean? Let's take baby one. So here we had mom and dad. Mom has a gene up here above the baby one, right? Same thing down here. There wasn't one there. Same thing with dad. There's not that one there, right? There's a big old blank space here. So these two genes are not present there. So the question is saying, why do you see genes in the parents that are not present in the babies? So in the chat box, let me know, why is there a difference between mom's DNA, dad's DNA, and baby's DNA? Let's see, Tim says, because the baby's DNA is a mixture of both parents' DNA. They don't inherit all of the genes from both parents. Excellent job, Timothy, right? We are not just clones of our parents, right? We are half mom, half dad, but we don't necessarily inherit 100% of our parents' DNA, right?
Yeah.
Remember that there is some independent assortment during meiosis, there's some mutations, there's that crossing over that happens. So the DNA is mixed up a little bit. You're still getting genes from mom and dad, but there is a lot of uniqueness to you that you're not going to necessarily be getting exactly from your parents. Very good, you guys. Awesome. You guys are doing great. So I think these questions are kind of fun, or these problems are kind of fun. The star doesn't usually ask them. Every now and then they do, though. I think it's kind of fun because you get to play investigator for a little bit. You get to pretend that, you know, you're not taking the star, but you're working at a police station. You're trying to figure out a case. Pretty interesting. Okay, so let's take our knowledge that we've equipped and let's go ahead and start practicing with some questions. So, Mrs. Smith has a baby named Tyra. She believes one of two men can be the father of her child. A paternity test is done and the results are shown above. Well, I guess it's below. Which of the two men are baby Tyra's father? So again, we have the baby's DNA, we have mom's DNA, and then these are the two different fathers. What we're trying to do then is figure out which one is going to be Tyra's father. So again, you got mom's DNA, you got baby's DNA. What you're trying to figure out then is which father is there. So I'm going to give you a minute. I want you to look through it. It's tough. We got dad 1 and dad 2 in the chat box. So let me know. It is a 50-50 shot. So far we're tied. Oh, dad 1's pulling through. Got about 20 more seconds. I think dad 2 might have the slight advantage. Okay. So let's look at dad 1 first. So look, you're looking for this one. Actually, we're going to use a different color. Let's use purple. So we got this one here. You move over to the side. It's kind of like in between these two. It's not really these. Okay, so you move on to the next one, and oh, I think we have a match. So that one seems like it might be a match to this one. This one seems like it might be a match for this one. You go down, not that one, not that one, not that one, not that one, not that one. Okay, so the only ones that are in contention for dad 1 are going to be those. Let's move to dad 2. We'll use pink. So not this one. This one could be, right? Could be that one right there. I think this one lines up with that one. I think that this one lines up with that one. This one lines up with that one. And there you go. Now, the reason some of these genes are going to be very similar is because sometimes genes are, you know, just that inheritable. So let's say, for example, we're talking about eye color or gene color. Maybe those are the same sizes between people. Hard to say, right? But yeah, I think it's overwhelmingly true that dad 2 is going to be the father. Even though dad 1 and dad 2 have some genes in common, I think it's pretty overwhelmingly true that dad 2 has more in common. Again, not the most scientific thing, but it's how it's done. Way to go, you guys. A lot of you got dad 2. And again, dad 1 did have some overlap. That is true. But we're looking at the one that had the most overlap. If you wanted to get really interesting and make this a story, the reason Dad 1 and Dad 2 might have such similar genes are because they're brothers, right? If they're brothers, it would make sense in that they would have very similar DNA. Not the same, but they might have some of those genes inherited from mom and dad, which would be similar, right? So pretty interesting. Okay, last practice problem. Lieutenant Russ is investigating a murder scene. The felon was scratched by his victim, and some of his skin cells were found under the victim's fingernails. A DNA test was performed. Which of the suspects is the murderer? So we've got DNA evidence that was taken from underneath the nail bed of someone who scratched the perpetrator. We're trying to match then that DNA to the person who committed the crime. Did you have a question, Miguel? No? Okay. All right, so I'm going to give you another minute on this one. So I'm going to give you a minute. Let me know in the chat box which of the suspects is the murderer. Oh, you guys got this pretty quickly. Okay, excellent, you guys. Wow, fantastic. You guys didn't even need the full minute. Right, so you're going through here. Let's use yellow again. This is going to be your biggest giveaway right here, right? This banding pattern, and or I guess really any of these right here too. When you're comparing S1, S2, and S3, they're all pretty unique, but S2 has these same patterns, right? And boom, that made your job real easy. So S2 then is going to be the perpetrator. Isn't that fun? So like I said, we can perform this experiment in the classroom. I can even take swabs of your mouth, cut it up with a restriction enzyme, run it out on a gel, and you can see your own DNA banding pattern. The fun thing about that is that your DNA banding pattern is going to pretty much stay constant throughout your life. So that DNA pattern is literally like your fingerprint. Very similar to the thing on your fingers, right? It's not going to go away. Your DNA is pretty much going to stay the same as you get older. So the DNA banding pattern goes with you, kind of like a biological identification card. Pretty interesting. Yeah, it's kind of it is really just matching. Now, when you get older and you decide to pursue this, maybe forensically, then there are a lot more wrinkles to it that make it a lot more math-based. You got to draw a lot of lines and use vectors to figure out the exact precise nature. A lot of stuff that we're doing here is simplified, so that you can learn it. But as you get older, as you do it, the process is not going to change. But the tools that you use to make sure that you are doing your job right will increase. Because again, this is usually a life or death kind of thing. And you want to make sure that you're doing it to the best of your ability and not just looking at the pictures and lining them up. All right. That being said, we do have an exit ticket. I think it's pretty simple. So I'm going to go ahead and copy that and put it into the chat box. Okay, so we've got a good 7 or 8 minutes to work on this, and actually more than that, because after this, what we're going to be doing is we're going to be working on assignment completion. So our goal for the rest of this is to be working on assignment completion. I'm going to go ahead and stop recording.